machinery. And if you talk about the replication machinery, replication, as all of you know, that replication is basically the formation of DNA from the DNA. And when the DNA copy is formed from parental DNA or from old DNA, that will be called as replication. And this process is accomplished by a variety of the enzymes and proteins. Collectively, those enzymes and proteins, they form the replication machinery. So if we talk about the replication enzymes, there are a number of the नहीं बंद है okay, there are a number of the replication enzymes which are performing their role in the process of replication. The first important enzyme is DNA polymerase. uh, DNA polymerase, as all of you are very much familiar with this enzyme, it it basically incorporates the correct nucleotide, or it copies the information from the template strand, and it incorporates those nucleotide into the growing polypeptide chain or nucleotide chain, and it polymerizes adjacent nucleotides to each other. So this is the function of the DNA polymerase. DNA polymerase is the major enzyme or the key enzyme which is involved in the process of DNA replication. By copying or using the information present on the other strand, this DNA polymerase incorporate new nucleotide into the growing DNA chain. The second type of the enzyme which is performing its role in the process of DNA replication, that is DNA helicase. As we have discussed in previous lecture, that the process of replication will always start from a specific point, and that is called ORIC, origin of replication. And at this origin of replication, the two DNA strands, they will unwind from each other, and when they will unwind from each other, for unwinding of these two strands, the DNA helicase will unwind these two strands. So the basic function of the DNA helicase is to unwind the DNA and melting of the DNA. Then we have another enzyme which is having its role in the process of DNA replication, that is called primase. Primase enzyme, it adds the primer, RNA primer, to start the polymerization. It will add the primer to start the DNA replication process, as without primer the DNA replication process, it cannot be started. So these are the major enzymes which are performing their role in the process of DNA replication. Then we have SSBP, single strand binding protein. As all of you know, that during the process of DNA replication, when the two strands, they will be separated from each other, they will unwind, and they will be separated from each other. So one strand of the DNA, that will be immediately, that will be occupied by these single strand binding protein, because if the single strand binding protein, they would not bind there at the isolated or separated DNA strand, that DNA strand can again rejoin some other DNA molecule, or it can form the complementary strand pairing with any other strand. So to stabilize the single stranded DNA, these SSBP, single strand binding protein, they bind, and they keep the DNA single strand after it has been melted by the helicase enzyme. So it is providing the stability to single stranded DNA. Then we have gyrase, another type of the topoisomerase. As all of you know, that topoisomerases, they sometimes they induce and sometimes they remove, or they relieve the supercoiling in the DNA. So DNA, this gyrase enzyme is basically a topomerase. It will release the torsional strand in the DNA molecule. Then we have ligase enzyme. This is another important enzyme. As we have previously discussed, that on the lagging strand DNA, the DNA is synthesized in the form of short fragments. Those fragments are called Okazaki fragments, and those fragments are gelled together. They are sealed together with the help of ligase enzyme. And then we have telomerase. Telomerase, it will finishes off the end of the DNA strand in eukaryotes, as we shall discuss in upcoming lectures, that sometime at the end of the DNA, the certain strand they tend to shorten after every round of replication due to presence of specific type of the sequences. Those are called telomere sequences. So this telomerase enzyme, it will compensate for the loss of the telomere sequences with the help of its enzymatic action. So these are some important enzymes which are having their role during the process of DNA replication. So if we look at this comparison or a tabulated form of these different types of the DNA A. This DNA A, which is basically a protein in nature, this is molecular weight and it is having one subunit. So the function of the DNA A protein is to recognize the ORI sequences. ORI sequences, as all of you know, that there are specific type of the sequences at which the replication process actually start. So that ORI, or origin of replication, that is having specific type of the sequences, and those sequences are recognized by this group of the protein that is called DNA A protein. Then we have DNA B. It is having a molecular weight 300,000. There are 6 subunits of this DNA B. It is also called as DNA helicase. This DNA B can also be called as DNA helicase, and it unwinds. The basic function of this DNA B protein or DNA helicase enzyme is to open the double-stranded DNA or unwinding of the double-stranded DNA. Then we have DNA C, another type of the enzyme or protein. 29,000 is the molecular weight. It is having one subunit and it assists DNA B to bind at origin site. So the basic function of the DNA B to recognize DNA A to recognize origin site, and the basic function of the DNA B to unwind the DNA at the origin of replication site. And the function of the DNA C is to it basically help the DNA B to bind at the origin of replication site and to unwind the double-stranded DNA. Then we have DNA polymerase. It basically synthesizes the new DNA strand. DNA G protein, which is also called as DNA primase. So this primase is having 60,000 molecular weight. It is one subunit and it synthesizes the RNA primer. As RNA primer is required to initiate the process of replication. Always remember that if primer is absent there, the process of replication cannot be initiated. So to initiate the process of replication, primer is always required. So this is function of the RNA primase, that it provides the primer, and this can also be called as DNA G. It can also be called as DNA G protein or DNA primase enzyme. It provides the RNA primer. Then we have single-stranded binding protein, SSB. There are a number of the single-stranded binding protein. Once the double-stranded DNA that has been unwinded by the DNA helicase enzyme, these single-stranded binding protein, they will bind to single strand of the DNA to provide stability. And DNA gyrase, as all of you know, that it is a type of the DNA topoisomerase, and the function of this DNA gyrase is to remove any type of the torsional strain or any type of the supercoiling in the DNA molecule. So if we look at the major enzyme which is performing its major role in the process of, can you hear me clearly?
Yes, sir.
Okay. So if we look at the DNA polymerase, which is a major enzyme, which is a key enzyme during the process of replication, and DNA polymerase of prokaryotes is it is also called as DNA dependent DNA polymerase 1. The first DNA dependent DNA polymerase or DNA polymerase 1 was basically Arthur Korenberg was a scientist. He discovered this DNA in 1958, and he later on in 1959 he received the Nobel Prize for the discovery of this DNA dependent DNA polymerase, which can also be called as DNA polymerase 1. So it can also be called as Korenberg enzyme, because Korenberg was the scientist who first time discovered this DNA. Later on, after the discovery of this DNA polymerase 1, certain other DNA polymerases, for example, DNA polymerase 3, DNA polymerase 2, they were also identified, and all of them, they are having the biological activity. I mean, all of them, they are performing the biological activity. And what type of the biological activity they are performing? They are performing 5' to 3' polymerase activity. Mean all of these 3 DNA polymerase, they are having the capacity to add the new nucleotide to the 3' end of the growing DNA chain. As I have told you that during the process of DNA replication, new nucleotide or deoxynucleotide, it will always be added to the 3' end of the growing polypeptide chain, as on the other end at the 5' end there is always phosphate group. So this DNA, all 3 type of the DNA polymerases, polymerase 1, 2 and 3, they are showing the some sort of the biological activity. And what is nature of that biological activity? That it will add the nucleotide from 5' to 3' end, and sometimes this DNA polymerase can also perform the exonuclease activity. What do we mean by exonuclease activity? This DNA polymerase can itself perform the exonuclease activity. Exonuclease is basically the proofreading activity. If there is any incorporation of the wrong nucleotide into the new DNA strand, this DNA polymerase will itself perform the proofreading activity, and it will remove that nucleotide, that is wrongly incorporated nucleotide. So this is, as all of you know, that exonuclease, they can cut the DNA on the sides, whereas endonucleases, they can cut the DNA from anywhere. So this is a comparison of 3 types of the DNA polymerases. DNA polymerase 1, 2 and 3. So if you look at their subunits, they are polymerase A, it can also be called as polymerase A. 1 can also be called as polymerase A. It is having single subunit. DNA polymerase 2 is having 7 subunits, and DNA polymerase 3 is having equal to 10 or more than 10. I guess this is more than or less than. So you will check this. I guess it is more than 10 subunits. This is for DNA polymerase 3, that can also be called as polymerase C or DNA E. 3 to 5' exonuclease activity. All 3 types of the DNA polymerases, they are also having the proofreading activity. Mean they can perform the 5' to 3' to 5' exonuclease activity. Mean it will start to read the newly synthesized DNA from its 3' end, and it will remove any type of the wrong nucleotide or wrong deoxyribonucleotide that has been incorporated into the new DNA strand. Then it is, if we look at the 5' to 3', only DNA polymerase 1 can perform the 5' to 3' activity. This is polymerization rate. Mean DNA polymerase can add 16 to 20 nucleotides per second. This can also be called as processivity of the enzyme. Similarly, polymerization rate of the DNA polymerase 2 or polymerase B, it is 40 nucleotide per second. Mean DNA polymerase 2, which can also be called as DNA polymerase B, it can add 40 nucleotide in a single second. And if you look at the polymerization activity or polymerization rate of the DNA polymerase C or DNA polymerase 3, it is 250 to 1000 nucleotide per second. They can be added by this DNA polymerase 3. Similarly, processivity mean number of the nucleotide added by the DNA polymerase to the growing polypeptide chain per unit time, that will be called a processivity. So DNA polymerase 1 can add 3 to 20 nucleotide, 3 to 200 nucleotide. DNA polymerase 2 can add 1500 nucleotide, and DNA polymerase 3 can add more than or equal to 500,000 nucleotide in a growing polypeptide chain. So this is what we can call it as processivity of the DNA polymerase. So these are the different properties of the DNA. So these sample are for either equal to or greater than. Okay. These are the polymerases of prokaryotes and specifically E. coli. So if we look at the exonuclease function, as I have told you that all three types of the DNA, they can perform exonuclease activity from 3' to 5' end, whereas only DNA polymerase 1 can perform exonuclease activity from 5' to 3' end. If you look at the 3' to 5' end exonuclease activity, it excites the mismatched nucleotide. For example, this in front of this A there should be T, but there is a question mark. It means any wrong nucleotide has been added. So this DNA polymerase will start to perform the 3' to 5' exonuclease activity, and it will remove this wrong nucleotide from this growing polypeptide chain. And only DNA polymerase 1 can perform the 5' to 3' exonuclease activity. Mean here in this case, it will start from proofreading from here, and it will remove any wrongly incorporated nucleotide into the growing nucleotide chain. DNA polymerase 1, this is the structure. It is basically responsible for proofreading, and it can also repair the DNA, damage area of the DNA. It can also fill the gap. So this is the function of DNA polymerase 1. Please ignore this heading. This is, I think, heading from previous lecture. Remove this from here. Okay. In this case, here you can see that this is N end and this is the C end. N end for nitrogenous end or amino end, and this is for the carboxyl end. This is the enzyme, and as all of you know that enzymes are basically protein. So protein are having amino group and carboxyl group. Proteins are made up of the amino acids, and amino acids are having the amino group and carboxyl group. So this is the N end of this enzyme or this protein, mean it is having amino group at this area, and this is the C end, mean it is having carboxyl end at this area. So when the DNA polymerase 1 it is cleaved, it will form the small, it will be forming the small fragments and large fragments. So small fragments will show the exonuclease 5' to 3' activity, and the large fragments, which is also called as clino fragments, they are having the DNA polymerization activity as well as it can perform the 3' to 5' activity. So that's why the two types of the DNA polymerase 2 and 3, they can show the all three types of the DNA polymerases, they can show the 3' to 5' exonuclease activity, but DNA polymerase 1 it can show only the 5' to 3' polymerase activity. If we talk about the DNA polymerase 2 function when the DNA polymerase 3 and 1 are not operative, not functional. If they are transitory, their function is arrested or their function is stopped. So the DNA polymerase 2 will take over the replication machinery. It can synthesize the DNA on the damaged template, and it can also repair the damaged DNA. It is also involved in the repairing of the DNA. So DNA, this is the function of the DNA polymerase 2. Then we have third type of the DNA polymerase. Yes.
सर कट गया
जी
सर आवाज कट के आ रही है
आपके नेट में इशू होगा सबको कट के आ रही है
आवाज बहुत कट
अभी फिर वो नेट में इशू होगा एक मिनट लेट मी चेक समटाइम नेट में प्रॉब्लम आ जाती है ना अब आपको ठीक आवाज आ रही है Can you hear me?
Yes sir.
अब ठीक है क्लियर है
Yes sir.
Okay.
Yes sir.
Okay. Then third type of the DNA, that is DNA polymerase 3. This is a heterodimer enzyme. Heterodimer means it is consisting of two different types of the subunits, and there are 10 different types of the subunits of this DNA polymerase 3, and it is showing the highest polymerase activity, mean 10 raised to the power 5 nucleotide per minute. As I have told you, this should not be minute, I think this is second, that 500,000 nucleotide per second can be added by this DNA polymerase 3. So it means this DNA polymerase 3 is performing the highest polymerization activity, and this is involved in the elongation process. The elongation process is majorly controlled by this DNA polymerase 3. So here you can see the structure of DNA polymerase 3. This is basically the hand-like structure in which there is palm, there are fingers, as well as there is thumb of the hand. So similar structure is for DNA polymerase 3. It is having various subunit. One is alpha subunit. Here you can see that there are number of the alpha subunits which are present in the DNA polymerase. Then we have epsilon subunits. There are number of the epsilon subunits. They are also there. So the function of the alpha subunit of DNA polymerase is 5' to 3' polymerization activity. Epsilon subunit has 3' to 5' exonuclease activity, and it also perform the role in the replication fidelity. Replication fidelity means that the replication process is free of errors, mean the chances of the errors or chances of incorporation of the wrong nucleotide, that is minimized by this DNA polymerase 3, and specifically epsilon subunit of this DNA polymerase 3. Then we have theta subunit. Theta subunit is involved in the maintenance of heterodimer structure. Here you can see that number of the different types of the subunit. For example, they can be 10 or more than 10. They are involved in the formation of DNA polymerase 3. Here you can again see that this is sigma. Sorry. This is sigma, this is gamma subunit, and this is beta claims or beta subunit. So variety of the subunits, they are combined and they are forming the structure of DNA polymerase 3. As I have told you that nucleotides will always be added to the 3' end. This is the 3' end, this is the 5' end. 5' end is always having phosphate group. So the 3' end is free, and this hydroxyl group, it will be attacked by the new coming nucleotide, and DNA strand has free hydroxyl group at the 3' end carbon or its terminal deoxyribose. So here at this point the new nucleotide will be added, and the process of replication will continue. Okay. Now we have, so in addition to these 3 types of the polymerases which are having their role in DNA proofreading as well as exonuclease activity as well as polymerization process or synthesis of the DNA, we have another type of the enzyme which is called DNA primase or RNA primase. RNA primase or DNA primase is an enzyme which provide the primer, which synthesize the primer. And what are the primers? Primer are basically the short RNA fragments of several nucleotides, 10 or more than 10 nucleotides. These are small fragments. Here this can be represented by the blue color. So primers are the small fragments of the DNA that are necessary to initiate the process of DNA replication. And this primer can also be called as DNA G primase. This primase enzyme can also be called as DNA G. Primase is able to synthesize the primer using free NTPs, nucleotide triphosphate, as the substrate and the single-stranded DNA as the template. So primer can synthesize small fragments of ribonucleotide or of RNA nature with the help of single-stranded DNA. So primase, it is involved in the synthesis of primer. This primer will provide the free 3' hydroxyl group to react with a phosphorus atom of the DNTP to form the phosphodiester bond. So this is the primer. Here you can see that the 3' end of this primer is free. So at this 3' end, new ribonucleotide DNTP deoxy ribonucleotide triphosphate will be added there, and hence the process of and DNA polymerase will continue the elongation of this single-stranded DNA. So it means the primase enzyme is involved in the first step where it synthesizes the primer. Once primer is there, our DNA polymerase 3 will elongate the strand from this primer. So primase, DNA B and DNA C and origin from a primosome complex at the initiation phase. So at the initiation phase, there is formation of a primosome complex in which the DNA helicase enzyme as well as primase enzyme, they will form a structure at the initiation phase. So another type of the enzyme are helicase. Helicases are also called as DNA B. It basically opens the double-stranded DNA, and it, as all of you know, that they are to open this double-stranded DNA apart from each other. Helicases will be requiring the ATP, and the opening process, this DNA or helicase carry out the process of unwinding of the DNA with the help of certain type of the other enzyme. For example, DNA A and DNA C. Then at the last we have single-stranded binding protein. Single-stranded binding protein, they are very much important in the sense that they are providing the stability to grow to single-stranded DNA, and single-stranded binding protein maintains the DNA template in the single-strand form in order to prevent the double-strand DNA formation, and it also protect the vulnerable single-stranded DNA from nucleases. So this DNA, it has two threats. One threat is that it is highly unstable and it can again form the complementary base pairing with any other single-stranded DNA. Another threat to this DNA is that this DNA is vulnerable to certain type of the nucleases. Those nucleases, they can attack on this single-stranded DNA and they can damage this DNA. So single-stranded binding protein, they actually protect this DNA from the destruction of the DNA exonuclease activity. When we talk about the gyrase enzyme, it basically removes the supercoil. If there are any type of the supercoiling in the double-stranded DNA, it will cut the phosphodiester bond and it will release the supercoil in the DNA, and it will again form the phosphodiester bond. It can change the DNA into negative supercoil state with the consumption of ATP. Then we have another enzyme which is called ligase enzyme. As all of you know that the function of the ligase is to connect to adjacent single-stranded DNA by joining the 3' end of the one DNA strand to the 5' end of the other strand. So hence the ligase, they are performing their role, and they can seal the nick, the gap which is present in the process of replication, repairing or recombination, or even in the splicing process, that can be covered, or that can be refilled by this DNA ligase enzyme. If we talk about the replication fidelity means high accuracy of the replication during the process. So replication, as all of you know, that it is basically the copying or the formation of new DNA strand from the parental DNA. It is base pairing during the replication is very much important. So enzyme basically they use two methods to ensure the replication fidelity or accuracy during the process of replication. One is proofreading and other is real-time correction or base selection. What is proofreading? Proofreading means, as I have told you, that DNA polymerase 1 has the function to correct the mismatched nucleotide. If there is any type of the mismatched nucleotide, whether present at the 3' end or the 5' end, DNA polymerase 1 will remove that unmatched nucleotide in the double-stranded DNA. So first function, how it actually performs this function, it identifies the mismatched nucleotide, remove this nucleotide by using its 3' to 5' exonuclease activity, and correct the base, and it will continue the process of replication. So this is all about the replication machinery, various types of the enzymes which are actually present during the process of DNA replication. If you have any question, you can ask, please.
सर आपने अभी threats बताए थे.
Yes. There are two threats for the singles. There are two threats. Two threats for single-stranded DNA. Once the DNA is separated from its complementary strand, it is highly unstable. So the chances are there that because it is having the necessary information stored in the single-stranded DNA, तो इसमें chances ये हैं कि ये दोबारा किसी दूसरे strand के साथ जो है वो base pairing कर देगा. एक threat ये है, और जब ये base pairing करेगा तो फिर replication का process continue नहीं रह सकता. Second threat is that कि अगर ये सिंगल स्ट्रैंडेड डीएनए है, इसके ऊपर प्रोटीन बाइंड नहीं कर रहे, तो इट इज वल्नरेबल टू द एक्सोन्यूक्लियस और द एंडोन्यूक्लियस, कि जो डीएनए को ब्रेक करने वाले एंजाइम हैं वो इस सिंगल स्ट्रैंडेड डीएनए के ऊपर अटैक कर सकते हैं 5' से या 3' से, और उसको वो क्या कर सकते हैं, दे कैन डिस्ट्रैक्ट इट. सो दीज आर द टू थ्रेट्स to which the single stranded DNA is exposed actually. जी, any other question? If you have any other question, you can ask, please. No question? Okay, ji, please stay connected so that I could upload your attendance on CMS. ओके जी, क्विज आपने फिर ईद के बाद देना है?
यस सर।
ओके, तैयारी करें। आपको मैंने असाइनमेंट्स भी भेज दी हैं, उसको भी तैयार करें, ठीक है? आयशा शफीक, आयशा, कैन यू हियर मी? आयशा शफीक, is she there?
Yes, sir.
Okay, then we have Kainat,
यस सर।
Selena, Selena, can you hear me? And we have Zoya Shah.
यस सर।
Okay ji. Okay ji, thank you very much. You may leave the session now. Allah Hafiz.
हाफिज़। हेलो, कोई है? कायनात?
मेरी आवाज आ रही है किसी को? हेलो, हाँ मैं हूँ। गुड खेली रह गई हूँ।